Hello, this is Andrew Wolf. In this video I'm going to talk about the pathophysiology of sickle cell disease. So this is a part of a series on sickle cell disease that I'm doing, and this is the second video. So in order to understand the pathophysiology of sickle cell disease, we need to think about the red blood cell, or the erythrocyte. So the red blood cell, a mature erythrocyte, is essentially a bag of cytoplasm that is filled with hemoglobin. There are no organelles in mature erythrocytes. All the organelles sort of get pushed out as the cell matures and after the hemoglobin is formed. And so it's just packed with hemoglobin that's dissolved in cytoplasm. In fact, if you dry out a red blood cell, 95% of the dry weight of a mature red blood cell is from hemoglobin. Okay, so, and not surprisingly, sickle cell disease is a disease of a deformation of hemoglobin. Okay, so here I've drawn a picture of one single hemoglobin molecule. The hemoglobin molecule is made up of 4 polypeptide chains that make up the one protein, and these have loose hydrogen bonding between them, so it makes a single protein molecule. And each chain of polypeptide has in the center of it, sort of held in the center of it, a heme pigment, and it's this heme pigment that bonds with oxygen and allows the hemoglobin to carry oxygen, which is the main function of hemoglobin, and the main function of red blood cells is to carry oxygen from the lungs to tissues for cells to use. Now, there's actually several different types of hemoglobin in the body, but the main type that adults have is a hemoglobin called hemoglobin A, which stands for adult hemoglobin. Now, there are 97% in a normal adult has of hemoglobin in a normal adult is hemoglobin A. There are other types. One is called hemoglobin F, which is fetal hemoglobin, and this makes up about 3% of normal adult hemoglobin, and obviously it's the most common type in fetuses, and it sort of slowly goes away and gets replaced by hemoglobin A during the first few months of life. Hemoglobin F becomes important when we talk about treatments for sickle cell disease, because one of the treatments actually involves increasing the concentration of hemoglobin F and replacing some of hemoglobin A with hemoglobin F using a medication called hydroxyurea. So we'll talk about that a little bit more. But I want to talk more about what hemoglobin A is. Hemoglobin A is made up of 4 polypeptide chains. There are 2 alpha chains. So there's 2 alpha chains, and there are 2 beta chains. And that's what makes up the 4 chains that make up the hemoglobin protein. Sickle cell disease involves a tiny mutation that causes a change in the beta chains of the hemoglobin molecule. In fact, it's one single amino acid. There's 146 amino acids that make up this beta chain. And one of them, in fact it's the 6th one, in the 6th position of the beta chain, gets changed from what is normally an amino acid called glutamate, and it is changed to valine. And this one tiny little change of one of the 146 amino acids in hemoglobin A changes the hemoglobin A into what we call hemoglobin S, and the S stands for sickling. So normal hemoglobin A usually stays nicely dissolved in the cytoplasm of the erythrocytes, but what happens is hemoglobin S has this valine molecule, and the valine molecule of one beta chain of hemoglobin S tends to want to stick to when it is in a deoxygenated state. So a bond forms, a reversible bond forms in between these 2 valine molecules, and what happens is these begin to form a little stack or a chain called a polymer. All right, so if you can imagine, this kind of continues, and we get, you know, this long chain, this long, straight, rigid chain of hemoglobin molecules that are all connected together. Now, so the hemoglobin is now not nicely dissolved, but it all sort of forms together into a long. So let's imagine what's happening inside the cell now. So instead of just having nicely dissolved hemoglobin, the deoxygenated hemoglobin S is forming these long chains that are connected together. And so they sort of form themselves into these long, what are called polymers. In fact, the process is called polymerization. So they've organized themselves into chains. So if you can imagine what's happening to the wall of the erythrocyte, it's getting stretched out into a sickle shape. So, and as these sort of form together, no longer do we have a nice round erythrocyte, but we end up with this sort of sickle shape with rigid rods or rigid polymers inside of it. Okay, so now we don't have nice round erythrocytes that can change shape and are pliable and can flow through narrow vessels. Now we have these long, rigid shaped erythrocytes that tend to get stuck in vessels. Okay, so I wanted to talk a little bit about what precipitates vaso-occlusive crises in patients with hemoglobin S. And a vaso-occlusive crisis, for those of you who haven't watched my first video, is when many of the cells sickle and clog up vessels, and as they occlude vessels, it causes ischemia and infarction to tissues that are distal to that occlusion. So you end up with sort of the stack of sickled cells. So what precipitates that? Well, we know that hemoglobin S, when it is deoxygenated, and I'm going to represent that with a bluish color here, if we have a significant concentration of deoxygenated hemoglobin S, then it is going to precipitate. But, so we'll imagine that this is a person with sickle cell disease, and we've got an oxygen saturation of in the veins of about 60%. So a little bit more than half of the hemoglobin S is still oxygenated. So that might not be enough to cause this to precipitate. What will cause it to precipitate is if the percentage of deoxygenated hemoglobin S increases, and that will increase the concentration. All right. So what will cause that? Well, obviously, decreased oxygen, right? So that can be caused by hypoxia. It can be caused by a patient that is short of breath, a patient that has asthma, a patient that has pneumonia. It could be a local decrease in oxygen from a decrease in perfusion. So we could have an area of ischemia. Now this can be precipitated by cold temperatures. So if your fingers are cold, that's going to cause vasoconstriction in your fingers, and it's going to decrease perfusion, and that is going to cause sort of local tissue hypoxia, right? And that's going to increase the concentration of deoxygenated hemoglobin S, and increase the chance that it may start to precipitate together and form polymers, right? Another thing that can cause an increased concentration of deoxygenated hemoglobin S is if we take this erythrocyte and we dehydrate it, so it now has a smaller volume, but it has the same amount of hemoglobin in, both deoxygenated and oxygenated hemoglobin S, and it's going to kind of smush all the molecules together and increase the concentration of deoxygenated hemoglobin S in the same space, right? So there's still the same proportion of oxygenated and deoxygenated hemoglobin S, but it's confined to a smaller area, and this also will make it more likely that the deoxygenated hemoglobin S will begin to precipitate and form polymers. And the last major factor to increase that cause polymerization is acidosis.